Restriction fragment length polymorphism. It is one of the field for DNA sequencing technology under the category of forensic science. Let's we start about introduction. Okay. RFLB is an enzymatic procedure for the separation and identification of DNA fragments. Each and every DNA fragments have a specific regions, have a different varieties between peoples. These regions are called as polymorphism. So, major principles of RFLB. RFLB is an enzymatic procedure. The desired DNA fragments would be restricted with the help of restriction endonuclease enzyme. The fragments is obtained from the desired DNA, which was detected by the use of restriction probes. Sudden hybridization using the restriction endonuclease enzymes for the isolation of desired length of DNA fragments is an simple are considered as important steps for RFLB. So, what are the procedures for RFLB technique? The first step is DNA extraction. DNA samples should be extracted from blood or saliva or any other samples and then purified. Second step is restriction digestion, and the purified DNA samples should be cut with the help of desired restriction endonuclease enzyme. These enzymes, restriction enzyme, have a specific restriction site on the DNA. So, it cut DNA into fragments. Maximum 4 to 6 base pairs of genes would be cut with the help of this enzyme. Gel electrophoresis. The digested fragment or cutting fragments are run in polyacrylamide gel electrophoresis or agarose gel electrophoresis to separate the fragments on the basis of their length, size or molecular weight. Different size of fragments from different bonds can be updated on the visualization site. Fourth step is denaturation. The gel is placed in sodium hydroxide solution for the denaturation. During the denaturation process, the double stranded DNA strands would be get to separate into single stranded form. Fifth step is blotting. The single stranded DNA can be obtained from the denaturation procedure. For this standard DNA can be transferred into charge membrane, that is nitrocellulose papers, by the process which is called as capillary plating or electroblotting. So, this is a diagrammatic explanation for the RFLB procedure. First we have to take a sample, then isolated a specific cells what we have to separate, and then purify the DNA fragments, purify the DNA sequences. Then with the help of the restriction endonuclease enzymes, the DNA could be digested, and the digested DNA fragments would be separated with the help of electrophoresis technique. Then it should be transferred to the sudden blotting or blotting technique. Then hybridization process will occur. After hybridization technique, the bonds will be observed with the help of autoradiograph. After that, the DNA sequencing could be done. Step 3, backing and blocking. The nitrocellulose papers can transfer the DNA is fixed by autoclaving. Then the membrane is waxed by using serum albumin or casein to prevent the binding labeled probe non-specifically to charge the membrane. With the help of these probe, we can easily find the DNA bands with the help of autoradiograph. Then last step is hybridization and visualization. The labeled RNA probes is hybridized with the nitrocellulose papers. The RFLB probes are complementary as well as labeled with a radioactive isotope, so that we can easily observe color bond under the visualization with the help of autoradiograph. So, next one is applications of RFLB. RFLB technique is most applied in the field of genome mapping, genetic disease analysis to detect the mutated genes, DNA fingerprinting. In genome mapping, it could be used to analyze their sequencing studies. The next one is genetic disorders. We can easily find the hereditary or genetic level diseases such as cystic fibrosis. We can easily find the disorders. Next one is DNA fingerprinting. For this, we can easily diagnose or find the paternity test or criminal identification with the help of DNA fingerprinting. Disadvantage of RFLB. So, RFLB requires a large DNA samples for the isolation and the separation of the desired fragments, and also time consuming process. In contrast, we can use PCR technique. We need only a small fragments of DNA and the less consuming process also. So now RFLB techniques is replaced with the PCR. The RFLB procedures requires also numerous steps and also takes weeks to yield the result. While techniques such as PCR, we can easily amplified with the target sequences within few hours. Thank you.